Hey guys, welcome back to a new video by Biology with Zhangxuan. So today we are going to discuss the Cambridge IGCSE Biology, Paper 4 Theory Extended, the 0610 Variant 2 of the October-November 2024 series. So as a disclaimer before I start the first question is that most of my answers in this video will be in point form. So make sure during your exam you need to write in a paragraph. And again, some of my answers here are just suggestive. Your answers can be right too. So please make sure that you cross-check with relevant resources to ensure that your answers are correct. Okay, so just keep on cross-checking with the relevant resources that you have. Alright, so again, this paper will be in the Google Drive, okay, and the description box, which I will put the link into. So let's start with the video. So question 1A. Organisms from the genus Chlorella are prototists. State the additional information required to name Chlorella according to the binomial system. So what can you see is that the genus is given, but they ask you for the binomial system. Binomial system, it contains the genus and it contains the species. So what is lacking here is the species. So like Homo sapiens, Homo is genus, sapiens is the species. So that is what you need in order to satisfy the naming system. Okay. Part B. Figure 1.1 shows the structure of an organism from the genus Chlorella. So part 1. Identify two cell structures in figure 1.1 that indicate that Chlorella is not a prokaryote. So it's just some features to differentiate between this organism and the prokaryote. And generally, you know that in prokaryotes, they have very limited organelles, which one of them is that it doesn't have a nucleus. Another one is that it also do not have a chloroplast. So you can just list there, nucleus and chloroplast is not seen, okay? If you put cell wall, I would say that that is wrong because cell wall actually is also present in a prokaryote, so be careful with that. Part 2. Identify two cell structures in figure 1.1 that are found in both fungi and prototists. So just find what are the similarities that are found there. So one of them is the cytoplasm and cell wall. Okay. Part 3. State the name of the two other kingdoms apart from the fungus, prokaryote, and prototist. So they have tell you that the three kingdoms have been listed. There's a total of 5. So what is the missing one? It's the common kingdoms that we see every day, which is the animal and the plant. Part C. Chlorella is sold as a nutritional supplement, and we also have spirulina, which is also another nutritional supplement. Table 1.1 shows some nutritional information between the two and the RDA for some of the nutrients. So I'm pretty sure you may have seen this question in your exam and then you may be a little bit shocked on how am I supposed to do this question. It's a little bit tough. It's okay. Again, this is a bit of mathematics here, but let's see what can we do. So one tablet actually contains 5 grams of chlorella supplement. So using the information in table 1.1, calculate the number of tablets of the chlorella supplement a person needs to take. Provide the RDA intake of iron and give your answer in 1 decimal place. Okay, so they've tell you that 5 grams of chlorella is inside 1 tablet, and there's also the RDA for iron. So we focus on iron, and the answer needs to be in 1 decimal place. Okay, so they have tell you that 37 milligram is found in per 100 gram of the substance. So we take 37 divided by 100. And again, don't need to change the unit. Just put as milligram divided by gram. Why? Because you're going to times by 5. 5 grams will cancel out both of the units and it leaves you with 1.85 milligram of iron. So now you know that in this roughly 1 tablet, there's 1.85 milligram of iron. So how many do you need to satisfy? How many tablets of this you need to satisfy? 12 milligram of iron per day. So you take 12 divided by 1.85, you're going to get 6.49 tablets, which is about equivalent to 6.5. You have to round off to 1 decimal place. So the answer is 6.5. Now, explain the advantages of taking chlorella as a dietary supplement rather than spirulina. So use the data in table 1.1 to justify your answer. So just look at this, okay, between chlorella and spirulina. What can you see is different? What can you see is adding? So see the values. There is vitamin C, there is calcium, but the iron do not change that much, but which we can actually ignore it. So we can just mention that it contains vitamin C, which the function of vitamin C is for immune system function, wound healing, and it also contains more calcium, which it helps to prevent rickets. So in this kind of answers, right, in this kind of question, that 4 marks, they ask you to explain. First, you describe what you can see from all comparison. Then you justify what is the function. That already consists 1 mark, 1 mark. If you can find out 2 comparisons and just list out 2 of its functions, each 1, 1. That's it, okay? Alright, part 3. State the name of one food that is the principal dietary source of vitamin C. This is very simple. If you've been eating your fruits every day, you should have known that orange is the one that has the most. But again, there are other answers, so just feel free to pop into the chat and just let me know, okay? For part D, chlorella is also a good source of protein. So they'll tell you again, there's a good source of protein. So what is the importance of proteins in active transport? So again, before we proceed, you know that active transport have protein carriers. If you remember protein carriers, you know what is its function is to help to move the ions against the concentration gradient, which we can see here. It's to produce protein carriers for the cell membrane. And what does it do? It links to the function, which is movement of ions against the concentration gradient, which actually corresponds to the definition of active transport, because they don't tell you what is the definition of active transport. So state it. What does it do? What does the protein carrier actually help contribute it? Okay. Now, state the name of the smaller molecules that proteins are made from. So when protein is being metabolized, what does it form? It forms amino acids. Okay, it's quite straightforward. Question 2a. Figure 2.1 is a diagram of a gas exchange system in humans. So again, it's a very familiar diagram from the past year papers. So I expect that you must know that you have to be able to label all of the parts of the lungs, okay, which have been stated in the syllabus, okay? So part 1, state the names of the parts of the label X, Y, and Z in figure 2.1. So what you can see is that X is pointing to the outside, okay, it's pointing to the outside, not the inside. So that is the external intercostal muscle. What about Y? So Y is linking to the smaller branches of the lungs, okay? This is like your 3 branches, okay? So this will be the bronchiole. And what about Z? Z is the U, it's almost like a U shape, okay? Maybe it's like a bended N shape. So that is always the diaphragm. Okay, the walls of the trachea contains rings of tissue. So state the name of this tissue and describe its function. This is something that you have to remember tight, okay? So it is the cartilage. And what does the cartilage do? It helps to prevent the trachea from collapsing. Part 3. State the names of 2 types of cells that are responsible for protecting the breathing system from particles. So this is quite straightforward. You know that this helps with your first line of defense. So what are the cells that are there? Ciliated cells or the goblet cells. Okay, it's quite straightforward. But of course you have to know these cells, these specialized cells. Okay, for part B, a scientist estimated the pressure and volume in the thorax during one breath. Figure 2.2 shows a graph of the results. So describe and explain the changes in the thorax occur during section A only. So they are referring you to this point and this point. So what happens here? So the pressure initially decreases and then it increases in the thorax. So the volume of air drops, but then later on it increases. And then what happens is that inspiration occurs, diaphragm contracts, and ribs move outwards. So the whole process in A, it shows that the lungs is actually undergoing breathing in process, or known as inspiration. Okay, And B, of course, will be expiration because it returns back to normal already. Okay. Okay, so this is a little bit of mistake. Pressure initially is higher, but then it decreases in the thorax because you just have to describe what you've seen from the graph. Okay, this is a little bit of a small mistake from my side. I apologize. Okay, for part C, okay, another type of fill in the blank question that you have to expect in the next coming exam series. So during physical activity, the CO2 concentration of the blood will increase because what happens is that you are constantly breathing in and out. So sometimes you do not have enough oxygen to replenish your muscles. So in the blood, there'll be much higher CO2. And this will be detected by the brain because the brain is the one that again it controls everything. Okay, controls all of your process. And what happens as a result? It increases rate and greater depth of breathing. Okay. Alright, question 3A. Figure 3.1 shows the kidney nephron and its associated blood vessels. Okay, again, it's a very familiar diagram that you should expect coming from excretion chapter. Okay, so the composition of fluids A, B, and C were analyzed. Table 3.1 shows the result of 5 components of the fluid. So they show you that again, this is very familiar that you can see here. And what can you realize is that initially it is glucose is present, but again it's gone at C. And then protein initially it has, but doesn't have it anymore. So you can see these are the things that you can focus on because these are something that is so familiar. Okay, so state the names of fluid A and fluid C. So fluid A you can see is this vessel. It it is going before the going towards the glomerulus. So that is actually the blood. And what is fluid C? Fluid C is the collecting duct, and this has already done all the absorption process that is supposed to be undergoing. As a result, it forms urine. So the answer is urine. Okay, for part 2, using the information in figure 3.1 and table 3.1, describe and explain the differences in the composition of fluid A, B, and C. So focus on these two that I've mentioned here. Okay, and then other than that is not so much, but you can focus on here. Okay, so they ask you to describe and explain. So this sort of question is very important to see. Okay, so you can see that on, only proteins, okay, of protein in fluid B and C, and 0 mg lah. Okay, there's nothing of protein in fluid B and C. So what happens is that it already undergoes filtration process. And what is the reason that B and C do not have protein? It's because that proteins are two large molecules. Okay, and there is also 0 milligram of glucose in fluid C. Why it's not in fluid C? Because all has been reabsorbed back into the body. And you can also see that there is also increased percentage of water in fluid C. And also most of the urea is found in fluid C also. So this is more of general description that you should provide to show your understanding of these things. Okay. So part B, outline how blood glucose concentration is controlled. So you can talk in a few different ways. When a person eats a lot of meat, a lot of good food, and the blood glucose concentration becomes high, what happens? How is it being controlled? You can also use this perspective. Or you can say the person is actually starving a little bit. And then what happens is that glucagon secretion is provided. Then you can see that glucose is being secreted into the blood. But let's use the good example with insulin, okay? So it is a form of negative feedback because negative feedback will link back to homeostasis. You want to maintain the constant internal environment. So what happens when the body is detected, it has a high blood glucose concentration, it is detected in the pancreas. So as a result, the pancreas will secrete insulin. Insulin will help to lower blood glucose concentration and they will help to convert glucose to glycogen and glycogen will store it in the body. okay, as like a storage form. That's it. So part C, a gene mutation may be involved in the development of type 1 diabetes. So describe what is meant by a gene mutation. So every time a definition question, it's always asking you to define. So you have to define this properly word by word, okay? So random changes in the sequence of bases in DNA. That's it. Simple as that. 2 marks. Okay, part 2. Outline the treatment of type 1 diabetes. So you should have learned in your syllabus there are actually multiple ways to treat type 1 diabetes. So I can give you some. Okay, one of my answers is a patient will have an insulin pump. Okay, because insulin pump has to be stick on the person's body so that insulin will be injected into the bloodstream. And this also helps to monitor blood glucose concentration. And of course, another treatment for type 1 diabetes is definitely to increase exercising, increase the physical activity of the patient so that at least you won't be able to solve type 1 diabetes, but you can alleviate the symptoms of it, okay, by encouraging physical activity to occur. Let's look at question 4. Figure 4.1 shows a diagram of 2 flowers from different plants of the same species. Describe what is meant by the term of species. This you can find in chapter 1, of course. So again, it's another definition question. You must nail this properly. So a group of organisms that can reproduce to produce fertile offspring. Okay, just simple as that. So complete the diagram in figure 4.1 to show self-pollination and cross-pollination by drawing one arrow to show the pathway of self-pollination and label this arrow, and drawing one arrow to show the pathway of pollen for cross-pollination and label this arrow cross-pollination, and again labeling the names of the structure involved in pollination. So first we can label the names. So this is the stigma, okay, this is the anther, and now we'll solve for self-pollination is that it comes from the anther to the stigma of the same plant. Same plant, automatically same species. Okay, so just draw an arrow like this. Well, for cross-pollination, it's the different plant, same species. So just draw a diagram from the an arrow from the anther to the stigma of another plant, but the same species. Okay. Now part 3. State the function of the parts labeled X and Y. So X is actually the ovary. So the ovary contains the ovule. And Y is the sepal. Okay, I just labeled this is the ovary, this is the sepal. So sepal will help to protect the flower. So these are just a very small things that you have to keep an eye on it so that you know this is actually a little bit tricky if you do not know what is sepal. Sepal just basically acts as a protection for the flower. Part B. Explain why self-pollination that results in production of offspring is a form of sexual reproduction and not asexual reproduction. So they tell you a clue that is sexual reproduction. So just talk about sexual reproduction alone. So first it involves gametes, which are actually in the haploid form. So what happens is that these gametes, before they actually can become haploid, they have to undergo meiosis. Meiosis is where they will divide the chromosomes from initially diploid to haploid. Then it involves fertilization because there will be fusion of the two gametes to form a zygote. And what happened? The offspring will show genetic variation because you are actually undergoing fertilization as a result. Okay? Okay, now for part C, state the type of environmental condition that hydrophytes are adapted to live in. So the theory is already hydrophyte. If you know what is hydro means water. So if you know this clue, just put aquatic because hydrophyte means they can live in water, which makes sense a little bit more, right? So yeah, that's why it's called as a hydrophytes for a reason. Alright, we have come to genetics. So question 5 shows figure 5.1 is a pedigree diagram showing the inheritance of a blood group in one family. So you can see that they show you this pedigree. You might have to go and look through it. It's actually by blood group. So again, blood group is a co-dominance. Okay, it's a co-dominance. Alright, So part A, state the number of people in figure 4.1 with XY chromosome. So XY chromosome means it's a male. So how many there are male? Just count. They have already given you the key. 1, 2, 3, 4. So the answer is 4. Only one IA allele. Okay, one IA allele. So 1, 2, 3. That's 3 IA alleles. Okay. Explain how figure 5.1 shows that blood group is an example of discontinuous variation. So this you have to know what are the features of discontinuous variation. So first they have limited number of phenotypes because you can see blood group, they only can show you blood group A, blood group B, blood group AB, and blood group O. There's only 4 you can get. That's why it's a limited number of phenotype. There are no intermediates, meaning there's nothing in the middle of it. It's just that if you get this, it means you are getting this. There's no sort of like middle where you're going to get something else. No, it is like that already. Okay? So state one example of discontinuous variation in plants. So seed color. So seed color is actually one example. Okay. And also the seed shape. Okay, it is also a form of a discontinuous variation too. It's something that you can't change. It's already set in your genetics. Okay. Part D. Explain why the inheritance of blood group is an example of codominance. So like I mentioned before, it is a codominance. So why? It's because that both IA, IB alleles will contribute to the phenotype. Okay, what does it mean? It means that when a human has IA and also has IB at the same time, there is a specific phenotype for them, which is actually blood group OB, blood group AB. So this phenotype actually exists. So you can see that blood group AB exists. So just give an example to show that your understanding of codominance is that if you have two different alleles, it will still show up something. It will not show you like, let's say it's IA and IB. In the result, they only give you IA. No, it doesn't work like that. It's not like a regular genetic inheritance. This is codominance. It means that both alleles can express something. Okay. So part E, a person has with the genotype IAIO has a child with a person with a genotype IBIO. So complete this genetic diagram and determine the probability. So you can see that parent 1 blood group A. So just match, okay? Just cross and do the Punnett square. Do your working here will be very good, very helpful. And you can see as a result, genotype is always the genetic makeup. So you must write in this form. Phenotype, you can write what kind of blood group is it. Is it blood group A? Is it blood group AB? And now, the probability. The questions have stated that what is the probability of having blood group AB? So you can see there's 1 AB out of 4. So you can see this is equal to 1 out of 4, which is 25%. Or you can put 0.25. That actually is also okay. Okay, that's actually acceptable. Alright, so this is very simple. If you know your Punnett square really well, this shouldn't be a big problem. Okay, we come to question 6. The flow chart in figure 6.1 shows one pathway of nitrogen as it travels through the nitrogen cycle and complete this flow chart. So 5 marks. It's actually really simple if you understand this entire process. So first, nitrogen in the atmosphere. You know that majority of the atmosphere is made of nitrogen. And nitrogen fixation by nitrogen fixing bacteria in the soil and also by lightning. Okay. Then uptake of nitrate ions and synthesis of proteins by the plant. And the plant is eaten by herbivores. Deamination occurs and they form urea and urea in the liver, and decomposition of animal and plant matter to form ammonium ions. The feces, okay, which has already been converted, will give out ammonium ions. Nitrification occurs to convert ammonium ions to nitrate ions, and the process of denitrification where the nitrates will become nitrogen gas again and goes to the atmosphere. So it's a story that you should know, but regardless whether it's in this form or in a 6 marks or 5 marks, you must be able to illustrate the story of the nitrogen cycle. All right. And part B, State the name of the two processes that occur in both the carbon and nitrogen cycle. So you just have to see whether you're familiar with the carbon cycle. So you have the decomposition, you have the excretion, because excretion in the form of gas, carbon dioxide, is also acceptable. And the last one, state the principal source of energy of input to the biological system. Energy input, and they say principal source. And obviously it's the sun, because the sun gives the most energy towards all of living things. Okay? So yes, that's all for this paper. So if you have any questions, please do comment in the comment section below. I hope you understand this video and all of the questions and the answers I provided. Alright, so see you in the next video. All the best. Bye bye.